for organizing this symposium. It has been a great opportunity for me to keep up with the latest developments in the computational drug discovery space, and also for us to present our contribution to great scientists all around the world. So today I'm going to present our work in virtual screening called Danvis, and I'm going to start with a question: is can you screen 1 billion molecules in a few days employing a data center? And I would like to show you how you can do it with the magic of deep learning and graph neural networks. So I'll go very quickly through the background, since I think most of you are very well informed. So on average, we know that the cost of getting a new drug into the market has skyrocketed over the past few decades. So pharmaceutical companies have started employing computers to help with various steps in the drug discovery pipeline. And one of them is virtual screening. We saw a lot of other works covering it, and that's also what we are doing. So in brief, virtual screening is when you have a protein and a drug, and you want to find for that protein, you want to screen a lot of different molecules and see which one fits the best in the binding pocket, so you can later use it as a drug. Yeah, so the most common way I've seen in the literature to approach this, and it was mentioned also quite a few times today, is so-called docking methods, where you have the protein and you know the binding site, and the algorithm gets a ligand and tries to fit it as well as possible to the binding site by rotating it, rotating its bonds, translating it in space, flipping it, etc. And that gives you a binding affinity score, and the pose of the ligand inside the protein pocket. There are also quite a few methods that build upon docking, and they use the output of the docking algorithm, the pose, to fine-tune the predicted binding affinity score using new data. And after you've done this, you have a lot of ligands with different scores, and you rank them, and you select the ones with the highest scores to move them along on the next step of the drug discovery pipeline. So one question here is what happens if you skip docking altogether? So what happens if you just take the ligand and the protein and feed them directly into a machine learning method and train them to predict binding affinity? And that's what our model does, and what we're going to show to you today. One approach to do this, at least. So the most obvious way to model molecules would be to think of them as graphs. Graphs are data structure that has nodes that are connected to each other with edges. And in the recent years, there has been a lot of development in so-called geometrical deep learning, where there's the introduction of the graph convolution, which is a generalization of the 2D convolution to graphs. So, for example, in images, you would have the 2D convolution here, where the node for this, the embedding for this node would be computed from all the adjacent pixels, whereas here you do the convolution with the nodes that are connected to the center one. For featurizing our inputs, we have two approaches. One which we apply to both ligands and proteins, the so-called atom level, and the surface level, which we only apply to proteins. For the atom level, you can think of a molecule, as I said before, as a graph with the nodes being its atom and the edges being the bonds between the atoms. And for each node, you can get a feature with some chemical properties of the atom, like the atomic number, the number of hydrogens, and that kind of thing. And the edges likewise can be given a feature vector by some chemical properties, like what the type of the bond is double or single, etc. For the surface level model, which we apply only to proteins, you first calculate the surface mesh using an algorithm that maps the protein to a 3D space. And here, instead of an atom, the node is no longer an atom. Basically, you instead have a 3D graphical object with polygons, and so it's vertex. Each node here of the graph is a vertex in this 3D object, and you can treat this thing as a graph as well. And in both cases, we don't give the entire protein to the algorithm, but only restrict it to the red area shown here, which represents the binding site. And as before, we can give each node some chemical features, and we also give it, in the case of the surface model, some geometric features that have to do with the shape and the curvature around that node. For our architecture, as we said before, we have the ligand and the protein. That those two go to separate parts of the overall model. There is a standalone ligand CNN that only models the ligands, and one for the proteins. After the graph convolutions are done, you get a ligand representation vector for that for the ligand that was inputted to the model, and likewise you get one for the proteins. The next step is to combine them with some function. We chose the outer product, but you can use other ways as well, like concatenation. And then you have a vector representation for the protein ligand pair. That representation can then be fed to a regression model. Again, you can pick your favorite one here, which went with the simplest one, which is the linear regression. And for labels, we give the binding affinity scores, whichever one is available from the dataset. This whole thing is trained end-to-end with backpropagation. Another important point is, as we said, we have two different models. The one with the atom level protein representation and one with the surface level protein representation. And we found combining them was really beneficial instead of doing prediction with just one of them. And we also employed ensembling. So this is when you train multiple models on the same data set multiple times with different initializations, so that you reduce the variance on the outputs. And this was also significant to performance. Okay. So now for our experimental setup, one of the most important issues in virtual screening is how to deal with biases, which are present in quite many data sets and have been reported across the literature. With this, we mean some underlying patterns in the data sets that a machine learning model can learn and can make predictions that match the training data without learning the protein ligand interactions that we are interested in. Instead, it learns something much easier. And if the training and testing share the same biases, we will get a high score on the test set as well, without actually learning anything useful for the real world. So one of my favorite examples of such biases are when, for example, the decoys are very easy to distinguish from the actives because they have different physical chemical properties like weight. So it's trivial for the model to tell them apart. But if you give it new ones, the prediction won't have any value. And so to mitigate this issue, we do two things. We have a separate training set from the two test data sets that are constructed by independent teams. And especially for the test data sets, which shows this is very well known one, DUD and LibCBA, both of them are specifically constructed to mitigate the bias issues. And also as a sanity check, we report the performance of a ligand-only baseline. So this is the same model as we saw before, but the protein part of the network is completely deactivated, so only the ligand goes through and is trained on the same way. So this would predict how good a ligand is on average without knowing the protein that it tries to bind to at all. And we would expect to be much worse than the protein ligand methods that take both into consideration. So a few words about our data sets. The PDB bind is quite widely used. It's constructed by selecting protein ligand complexes from PDB, the database that has all the 3D structures of proteins, that also report the binding affinities. And it consists of two different sets, the refined and the general. General also contains all the complexes that are in refined, and the refined is basically has stricter selection criteria with respect to the quality of the 3D structures. And for testing, we use DUD, which as you know, probably has about 100 proteins, and for each active, they generate synthetically 50 decoys with similar physicochemical properties, but different to the topology. And another testing dataset that has been increasing in popularity is LibCBA. In this case, the inactives are not generated, but instead they are experimented, validated experimentally. And the actives have potencies that are typically found in screening decks, which are usually quite lower than the ones you see in PDB bind. Okay, here there is a problem with the PDB bind dataset. If we train with just that, we only have 3D complexes that contain true binders, and so the model only sees positive examples with a given binding affinity score. The problem with that is that the model never sees a decoy, and so it won't be able to discriminate between actives and decoys on the new dataset that you try to run it on. So to solve this problem in the training set, we did negative sampling, perhaps the most important component for this algorithm to work. So the idea here is that one protein is active can act as another protein's decoy. So imagine this is your original training dataset, like PDB bind. You have a protein with a ligand that give a binding affinity score of 7, but you can take the ligands for two other proteins and give them a negative binding affinity score of 0, labeling them as negatives, essentially. So this does indeed introduce the possibility of ligand 2 being an actual binder on protein 1, but well, this is very hard to happen because if you select an active a ligand at random, it's very unlikely that you will it will actually bind to your protein. Otherwise, we wouldn't need to do virtual screening at all. So yes, we introduce some noise to the algorithm, but the overall effect of the negatives is much more important than the noise we introduce. So let's have a look at our results. And before we do that, a quick word about metrics. We report the area under the curve metric. So this is a standard classification metric. It is used for classifiers all across the machine learning literature, but it's not ideal for screening because in screening you only really care about the very top of your predictions. So in order to quantify how well we will do in screening, we use screening specific metrics like bedrock and the enhancement factor. Bedrock is like a modification of the area under the curve, but it exponentially weighs the data points at the top of your predictions most. So you still get the whole overall performance of the classifier over your entire data set, but you pay more attention to the top of your predictions, whereas the enhancement factor only looks at a given percentage of the top rate predictions, and it's like the recall at that percentile, and which are the 1%, which is commonly reported. Yeah, but it does not take at all into account anything that is not in the top 1%. So here are our results. We present our models here in these two light blue colors. Danvis G is trained on the general set of the PDB bind, and Danvis refined is trained on the refined set. This one in orange are commercially available solutions that also do virtual screening. VINA is the most common docking algorithm, and these three with green are methods that build upon VINA, like the ones I mentioned before, that you have machine learning methods that take the output of the docking as input to calibrate the prediction score. So if we look at the screening metrics, we see that our Danvis general model is doing significantly better than all the competition, at least on this data set. Although I'll have I will mention that Nina itself was trained on the refined set, so the most fair comparison would be with Danvis on the refined set, which is still very competitive with the rest of the methods. And some methods even do worse than the ligand-only baseline. Yeah. So, and for the next benchmark, this one appears to be much harder. All the scores are very much lower. The ligand baseline starts looking more competitive. It is a newer data set. There is not a lot of published work in it. That's why we only saw these ones that we were able to find. And one possible explanation is that is due to the lower potencies of the actives in this data set. Yeah, and I hope the community will go on and figure out how to score higher in this data set as well. We are also investigating. All right. So for I wanted also to include then the ablation study we did, which quantifies the effect of different parts of the pipeline we used on the overall outcome. And as I mentioned before, negative sampling is by far the most important component. You can see here with pink, if you train the model without negative sampling only on the positive examples, the enhancement factor shrinks significantly. And also another important component is the ensembling. So if you compare the orange one to the green one, you see there is a big gain as well from doing multiple runs of the same model. And finally, I want to highlight the impact of the combination of the atom level and surface level modalities, as well as you can see here. The individual modalities performances are much worse than the combined models. And this behavior that I mentioned is replicated across all the metrics. So I teased you before a bit about the screening times. So here are some numbers. Danvis is about 1,000 to 10,000 times faster than the competition, depending on which competitor you choose. So it's 10,000 times faster than docking, and yeah, this speed is because it leverages the very highly efficient algorithms for linear algebra that are done on GPUs, and the whole deep learning ecosystem is built upon. And also you can do a trick to speed it up even further by storing some embeddings into the memory. So regardless of which protein you choose and which ligand, the embeddings will be the same for those particular ones, regardless of the pair. So if you have, let's say 1 billion ligands and 10 proteins, you can first compute the embeddings for the ligands and only combine the embeddings of the protein, and that means you only run the final regression model at the end, saving a lot of time. And yeah, faster, of course, means better and cheaper. Well, we say that because the 3 orders of magnitude is a significant advancement in speed. With shorter screening times, you can screen a lot more molecules, and hopefully that means that you will include a highly active ligand in your search space that you will actually find with the algorithm. Great. At this point, I would like to thank the whole DeepLab team, and especially the bioinformatics subteam for their contributions in Danvis and other bio projects. I would also like to give a huge thank you to previous colleague and mentor, Agamemnon Krasoulis, who was integral to the development of Danvis. A few words about us. We are a startup in Greece founded by academics. We specializing in machine learning, and our goal is in general to bridge the gap between academia and industry. Apart from the bio team, DeepLab includes a much broader team of talented researchers and engineers in deep learning. We have over 15 years of experience in deep learning with a variety of academic and large-scale R&D projects in areas like recommender systems, computer vision and language, neuroinformatics, and we started also branching out into the health applications recently and bioinformatics, and inspired by our expertise in the previous domains, we now run projects in collaboration with external collaborators in cancer subtype prediction, handling RNA single-cell data, utilization of cell-to-cell communications and multi-omics, and target identification. All this with our knowledge in a variety of machine and deep learning methodologies. Yeah, so to conclude, we've shown our virtual screening algorithm, Danvis, which is much faster than the competition, achieves significantly better performance than the commercial-based competitors in at least the benchmark, and is on par with the competition on the PCBA dataset, and while being 1,000 to 10,000 times faster. And I would also like to mention we are very active in biohealth related areas, and we seek further collaborations to advance the scientific research towards practical results of high impact, so we can best utilize the powers that have been given to us by the deep learning and machine learning advances. That's it for me. Thank you very much for your attention, and I would be very happy to take up questions.